Hello everyone, welcome to the world of me and microbes. Me, Ankita Patadia, Assistant Professor in Microbiology Department at LJ Institute of Applied Science, LJ Campus, Ahmedabad, and about the microbes. Today's our discussion's topic is mode of nutrition. Nutrition is required by all living entity for the survival, and the microbes are having different mechanism for the uptake of the nutrition. So in this session, we will study how the microbes will get nutrition and what is the mechanism, how it occurs. We will see in the detail about the topic in this session. Let's begin the discussion. The topic is from the syllabus of Gujarat University, BSc Semester 3, Core Course MI 201, Microbial Physiology, Unit 3, Microbial Nutrition and Introduction to Metabolism. So let's begin with the introduction, how the nutrition occurs. All the living cells are exclusively dynamic entity, and that cells are separated from the environment by a semi-permeable cell membrane, or membrane is there which will separate the cellular environment from the outer membrane in a carefully and controlled manner. Here, that cell membrane will allow only some particles to move inside. That's why there is semi-permeable, and sometimes some molecules can move and sometimes some molecules cannot move. Even sometimes some molecules can move inside simultaneously or together inside the cell. So different mechanisms are there for the transport of the various molecules inside the cell via permeable membrane, and according to that controlled manner we can classify the nutrition uptake. Nutrition uptake is classified mainly first in two classes, that are passive transport and active transport. Now that passive transport is again divided in three reactions, that are simple diffusion, facilitated diffusion and osmosis. These three reactions combinedly considered as a passive transport of the nutrient. And in active transport, first is primary active transport, that is having one type of reaction, role of binding protein. Secondary active transport, that is having three types of reaction, that is symport, antiport and uniport. Now third reaction in active transport is phosphate bond linked transport, and the fourth is group translocations. So this is quite interesting topic. So today we will start our discussion with the passive transport. So in this session we will discuss only passive transports. That why I have given the name as a part 1 over here. So active transport we will discuss in our next session. So begin with the passive transport over here. What is the passive transport? It is a passive transport means direct transport of the nutrient molecule via cell membrane without utilizing energy. So three types of passive transport mechanisms are there: simple diffusion or passive diffusion, facilitated diffusion and osmosis. Let's start the discussion with the passive diffusion and followed by facilitated diffusion and osmosis. Before that, what is diffusion we have to understand. Diffusion, we can see in this picture, three jars are there having conical flasks open and jars are sealed. That conical flasks are having gaseous material, substances are filled inside. So in very first jar we can see that substances are coming out from the flask to inside the environment of jar. Here more molecules are came out, and here all the molecules are equally distributed. So diffusion means movement of molecule from the region of higher concentration towards this lower concentration. So the molecules are in high concentration in the area of flask and they are moving towards the lower concentration, means area of that jar. So movement of this molecules from higher concentration towards the lower concentration is called diffusion. Now diffusions occur in the concentration gradients, means from higher to lower. In cell membrane is having two sides, opposite of the cell membrane and the inside the cell, two different sides. The concentration of the molecules are different, and concentration gradients means from higher to lower. So here diffusion occurs till the equilibrium gets. Here we can see that in third jar equilibrium is there. Up till that, starting from the first jar to second jar, this is a continuous process showing jar, not three different jars are there. First jar, after some time the picture of that first jar, and after some time here we can see that equilibrium occurred and the diffusion is finished. Here, this conical flask is covered with the semi-permeable membrane. So this is the example of diffusion. Now starting with the, here diffusion is the occurs because of random movement of this particle. Because the movement of the particle not in a particular manner, they are diffusing in a random manner. So begin the discussion with passive diffusion. We have now understood what is diffusion. In passive diffusion, molecules move from region of higher concentration to the one of lower concentration. It occurs it occurs in a concentration gradient direction, and it depends on the size of concentration gradients, as we have discussed, from higher to lower. It does not utilize any form of energy for the diffusion, so it is not consuming any energy for the transport of that molecule. And diffusion is affected by the size of the particle, charge of the particle, as well as the charge of the on the polar surface of the cell membrane. Now, which molecules can pass through this mechanism? Steroids, some gases can passes via passive diffusion from cell membrane rapidly. Now, various factors that affect here, water soluble molecules such as the hydrogen ion, concentration ion, sodium ion, they also can, small molecules can pass through the membrane via passive diffusion. In this picture, we can see that small uncharged molecules are there in a higher amount in first portion. After some time, this portion is like this, and after some time, this, let's understand, this is the bilayer plasma membrane. So initially, that smaller part will outside the cell, so they will move inside the cell via permeable membrane to the inside the cytoplasm. So from the extracellular fluid, this molecule will come inside till the equilibrium gets here. So time is directly affecting the diffusion process, as well as other factors also affecting, that are temperature, means the temperature is high, the rate of passive diffusion is high. Solvent density, if the solvent is very dense, then the diffusion process will become slow, or passive diffusion becomes slow. Now other factor is mass of molecules. As I have said that smaller molecule diffuse quickly compared to a very bigger molecule, and extent of concentration gradient means inside the cell, the total area of the cytoplasm, means the size of the cell is high, the passive diffusion required more time. So this is all about passive diffusion. Now second is facilitated. In facilitated diffusion, it requires some carrier molecule to make the entry inside the cell of the molecule, means carrier molecules are required, or carrier proteins are required in the cell membrane for the transport of smaller molecules. Here carrier proteins are sometimes called permeases, means they are permeating smaller molecules to come inside the cytoplasm, which are embedded in the plasma membrane, means they are not externally, but they are inbuilt in the plasma membrane. This Facilitated diffusion is driven by concentration gradient. As passive diffusion, it also occurs in concentration gradient. Now, that carrier protein sometimes works as an enzyme, means they are specific to the substrates. Each and every carrier molecule not allow all the particles to come inside or all the molecules to come in cytoplasm. So, specific carrier proteins are there to make availability of some specific nutrients to the cell. Each carrier is selective for the transport of only closely related molecules. Now that energy is not required in this reaction. In passive diffusion also energy is not required. Both the reactions are reversible, means concentration gradients will affect. If the concentration of the molecule increase in the cytoplasm, they may come out of the cytoplasm to the outer environment via that permeable membrane. So both the reactions are reversible reaction. Here in the picture we can see that these molecules are coming inside the cell from the outside via with the help of this carrier protein, which is also called as a permeases, or it only allows specific molecules to come inside. Here we can see that molecules are coming inside. This is the reversible reactions and doesn't require any energy. So if the concentration of the molecules will become increase here, they can come outside of the cell as well. Now third is the osmosis, special case of diffusion in which solvent molecule diffuses across the semi-permeable membrane. In passive diffusion and in facilitated diffusion, solute molecule moves via permeable membrane, while in osmosis, solvent molecules moves via permeable membrane. In the picture, we can see that the solvent molecule move from low to high solute concentration, means high solvent concentration, low solvent concentration to high solvent concentration. Here we can see that the solute here is in a low concentration and here in a higher concentration of the solute is present. So solute molecules are more, here solute molecules are lesser, means water molecules both the side equal, but the comparatively here the water molecules are lesser. So from this, with the via this semi-permeable membrane, this water molecules come this side because here the ratio is not equal, means equilibrium is not there. So this only 6 molecules of water, solute we can see that, and water molecules are higher. So this higher number of water molecule comes this way. So we can see that the solvent is works as water is works as a solvent, and the movement of the solvent molecule from the higher concentration towards the lower concentration. So both the solvent and solute comes in equilibrium over here. So this is a very special case. Now osmotic pressure is defined as the pressure to require or to prevent the net flow of water by osmosis. If we want to prevent this process, we require some pressure, and that is called osmotic pressure. Now, osmotic pressure is very well example described by tonicity. We can see that how the water molecules come inside the cell. The example of this balloon and the beaker having different solutions. This is the balloon or having a permeable membrane balloon. We can see that here 3 beakers are there having different solvent and solute concentration. Here 1% salt concentration, weight by volume, here 0.5% salt concentration, and here 3% salt concentration is there. Now in the cell, this is the cell permeable membrane. Here 1% salt concentration present in the cell, so no movement of any ion. This is called isotonic solution. Now here the solvent molecule is comparatively higher outside the cell membrane, means 0.5% salt concentration is there, which is lesser than the 1% present inside the cell. So here the water molecules will come out, and here third is showing 3% of the salt concentration and 1% salt concentration inside the cell. So the more amount the salt concentration present in the outer environment, so water molecule will come inside the cell. Here the cell will rupture, here the cell will shrink. So this different osmotic pressure creating environment is there. We can see that in the osmosis pressure. Now confusion occur in the osmosis and diffusion. So let's remove this confusion here. Here we can see simultaneously two reactions, diffusion and osmosis. So it will help in removing the confusion. In diffusion, the solute molecule will diffuse, and here in solvent molecule will diffuse. Here the high solute concentration, so it will diffuse and it will till the equilibrium get. So here is the diffusion process. Solute molecule will diffuse in the medium. Here the semi-permeable membrane is there, and water molecule means solvent molecule will move through the semi-permeable membrane. Here no membrane is there. Diffusion means free or random movement of the molecule or particle is there. So now the confusion might be clear about the diffusion and osmosis. Let's summarize each three reaction in this figure simultaneously. First figure is showing that plasma membrane. Here the outside the cell solute molecules are more, and they are coming inside the cell through the plasma membrane via simple diffusion, no energy, no carrier protein, and completely on concentration gradient base, and this is the reversible reaction. Second is showing that non-specific transporter. Here carrier molecules are of two types, as I said, specific as well as normal protein carrier molecules. So first figure is showing that non-specific transporter is transporting some molecules inside the cell from the outside in concentration gradient. No energy is required, and this is also a reversible reaction. And in C picture, we can see that specific transporter or specific carrier molecule is there present, which allow only specific molecules to come inside. Different molecules are there, but only allowing specific molecules to come inside the cell. Here the energy is not required. And the third is osmosis, means movement of solvent molecule from the outside to the inside the cell. So I hope you understand all these three reactions in detail, and these reactions are not utilizing any form of energy. So that's why they are called passive transport of the nutrients. So this is the list of references which I have been used for the discussion of the topic. Thank you.